Your hands are identical in structure, yet impossible to superimpose, a property known in chemistry as chirality. This spatial arrangement defines the behavior of organic molecules within the human body. Life is homochiral, meaning our biological systems are built from specific spatial orientations, such as left-handed amino acids and right-handed sugars. When a drug molecule enters this environment, its shape determines its fit into cellular receptors, like a key in a lock. A single misplaced atom or a mirrored orientation can transform a healing substance into a potent toxin. This geometric reality was the hidden driver behind one of the greatest medical disasters in history. In the late 1950s, a sedative named Thalidomide was released to the public, marketed as completely safe for pregnant women. However, the manufacturers overlooked a fundamental rule of stereochemistry that would lead to thousands of severe birth defects worldwide. Thalidomide was synthesized and sold as a racemic mixture, containing equal parts of two mirror image molecules called enantiomers. Chemically, these are labeled as the R+ and S- forms. While they share the same molecular formula and connectivity, their three-dimensional arrangement differs at a single chiral center located on the glucuramide ring. In a laboratory setting, these two forms often exhibit identical physical properties like boiling points or solubility. However, the human body is a chiral environment that treats these two shapes with extreme distinction. The R-enantiomer provides the intended sedative and anti-nausea effects by interacting with specific neural pathways. Unfortunately, the S-enantiomer is teratogenic, meaning it interferes directly with fetal development. At the time of its release, pharmacological testing did not require the isolation and study of individual enantiomers, assuming that the biological activity of the mixture was simply the sum of its parts.
Even if scientists had successfully isolated the helpful R-enantiomer for clinical use, a deeper chemical trap awaited. Within the physiological environment of the human blood, at a pH of 7.4, thalidomide undergoes a process called spontaneous racemization. This occurs through keto-enol tautomerization at the chiral center. The acidic hydrogen atom at the alpha position of the glucuramide ring can be lost and subsequently regained from either side of the molecular plane. This reaction creates a planar intermediate that effectively flips the molecule between its R and S configurations. Within hours of ingestion, a pure dose of the helpful sedative transforms into a dangerous 50-50 mix of both mirror images inside the patient. This rapid interconversion meant that thalidomide could never be safe in its original form, as the body itself acts as a catalyst for creating the toxic version of the drug. The destructive power of the S-enantiomer stems from its ability to bind to a protein called cereblon. This protein is part of an E3 ubiquitin ligase complex, which typically identifies and marks specific proteins for degradation within the cell. When thalidomide binds to cereblon, it alters the substrate specificity of the enzyme. Instead of its normal targets, the complex begins to destroy transcription factors that are essential for limb development and organogenesis. One critical target is the protein SAL4. By forcing the premature degradation of these developmental blueprints, thalidomide halts the growth of limb buds in the early stages of pregnancy. This molecular hijacking happens with surgical precision, showing how a minor shift in atomic orientation can reconfigure the entire protein management system of a cell, leading to the devastating physical malformations observed in thalidomide victims.
Beyond protein degradation, thalidomide interferes with the formation of new blood vessels, a process known as angiogenesis. The growing embryo relies on a highly coordinated expansion of the vascular system to provide nutrients to developing tissues. The S-enantiomer inhibits the growth of these vessels by downregulating vascular endothelial growth factor. Without an adequate blood supply, the rapidly dividing cells in the limb buds undergo oxidative stress and apoptosis, which is programmed cell death. This explains the specific patterns of limb shortening, known as phocomelia, where hands or feet are attached close to the torso. The chemical interferes with the signals that tell the body where and how far to extend a limb. Understanding this anti-angiogenic property later opened new doors for medicine, but in the context of a developing fetus, it was a catastrophic disruption of the biological timing required for normal growth. A common misconception in mid-20th century medicine was that the placenta acted as an impermeable barrier, protecting the fetus from most drugs and toxins. Thalidomide shattered this assumption. Due to its lipophilic nature and small molecular weight, the molecule crosses the placental barrier with ease through simple diffusion. Once in the fetal circulation, the lack of mature metabolic enzymes in the developing liver means the fetus cannot efficiently detoxify or clear the drug. The concentration of the S-enantiomer remains high in the embryonic environment during the critical window of day 20 to day 36 after conception. This specific time frame is when the heart, ears, eyes, and limbs are forming. Because the drug persists in the fetal tissue, even a single dose taken by the mother during this narrow window was enough to cause permanent and profound developmental changes.
While thalidomide was being distributed in over 40 countries, it faced an unexpected hurdle in the United States. Dr. Frances Kelsey, a reviewer for the Food and Drug Administration, refused to approve the drug despite intense pressure from the manufacturer. Her skepticism was not based on the birth defect reports, which had not yet surfaced, but on data concerning peripheral neuritis in adult users. She noted that the safety data provided was insufficient and lacked rigorous evidence that the drug did not cross the placenta. Her insistence on thorough pharmacological data prevented a large-scale disaster in America, highlighting the vital role of regulatory oversight. This standoff demonstrated that chemical safety cannot be assumed based on anecdotal evidence or lack of reported side effects, but must be proven through controlled studies that account for different physiological states, including pregnancy and long-term use. The global fallout from thalidomide led to the 1962 Kefauver-Harris Amendment, which fundamentally changed how drugs are tested. Before this tragedy, manufacturers only had to show that a drug was not toxic. After the amendment, they were required to prove both safety and efficacy through well-controlled clinical trials. This birthed the modern multi-phase clinical trial system we use today. Furthermore, the event forced the scientific community to develop specific protocols for teratogenicity testing, requiring drugs to be tested on multiple animal species during pregnancy before human trials. It also brought the concept of stereoisomerism to the forefront of drug design. Regulatory agencies now demand that every enantiomer in a mixture be tested individually as a separate chemical entity. This ensures that the distinct biological pathways of mirror image molecules are understood long before a product ever reaches a pharmacy shelf. Paradoxically, the very mechanisms that made thalidomide a tragedy have made it a powerful tool in modern medicine. Today, thalidomide and its derivatives, known as IMiDs, are used as primary treatments for multiple myeloma and certain complications of leprosy. Its ability to inhibit angiogenesis is utilized to starve cancerous tumors of the blood supply they need to grow. By targeting the cereblon protein, scientists have developed new drugs that can specifically degrade disease-causing proteins that were previously considered undruggable. This field, known as targeted protein degradation, is one of the most promising frontiers in oncology. However, the use of these drugs is now strictly controlled through systems like the System for Thalidomide Education and Prescribing Safety. This ensures that the drug is never administered to anyone who is or could become pregnant, turning a historical poison into a controlled, life-saving therapeutic agent. The story of thalidomide remains the ultimate case study in the power of molecular geometry. It taught the scientific world that in chemistry, structure is function, and even the smallest spatial variation can have life-altering consequences. We now treat chirality as a primary design consideration in everything from pesticides to perfumes. Modern synthetic methods, such as asymmetric catalysis, allow chemists to produce pure single enantiomers, avoiding the risks of racemic mixtures entirely. This precision ensures that the drugs of the future are more effective and carry fewer side effects. As we continue to map the complex interactions between molecules and the human body, the lessons of thalidomide serve as a permanent reminder of the necessity for rigorous testing and the profound impact of three-dimensional symmetry in the microscopic world that governs our existence.